We are a global organization looking into gene and cell therapies especially in the US. We are about to submit a couple of INDs for a gene therapy and also for a cell therapy. And as a company, we have a lot of experience with something that is really changing the way we see the world of healthcare now. And this is, again, this revolutionary approach to gene and cell therapy. What does it have to do with an European medical device investigator-initiated study and virtual conference? So I'm going to tell you about that. First, my background. I was originally born in Spain. I thought that was important here. I spent many years in Germany, so I'm very familiar with the European environment. I worked for Abbott and AbbVie for over 15 years. I've been working also with other companies. And in the past 10 years, again, I've been focusing with biotechs in this space. Now, when thinking about devices, that's something that someone that has worked in Abbott Labs, I've done for many years, and I'm going to give you some examples. And also my background is in immunology, so I work in academia and clinical research in that space. So I would like also to show you how important is when we work with investigators to ensure that they not just conduct their study, but they also look into biomarkers. And they're many times important, I think, also when you use medical devices and also any type of biopharmaceutical. Next slide, please. So this is an outline of what I'm going to be discussing. First, when talking about biomarkers, I think I would like to imprint in your minds the topic of personalized medicine, that we're moving into an arena where patients are treated more and more based on a number of signatures that are either genetic or are related to other factors, demographics, and so on, that are going to really individualize the type of therapy that we're giving to these individuals, these patients. So I will show you an example of a drug and medical device combination product that I was working on. And also, I, again, will emphasize on the topic of biomarker-driven clinical trials. Then I will show you, let's call it 2 case studies, which are the areas I'm focusing on right now, treatment of hemophilia A and how we select investigators for our studies, and cell therapy also, and how we do that in a very serious condition called graft-versus-host disease happening in patients that receive bone marrow transplantation. Next slide, please. Next. So if I have something I call claim to fame, at least internally for me, is the work I did at Abbott AbbVie. At that time was still Abbott Pharma, moving into AbbVie. And this is being on the development team for the Humira Pen. The Humira Pen was absolutely revolutionary in the mid-2000, about 2005 or 6. And we at Abbott just took the example of the insulin pens, and we developed one that would adapt for patients with rheumatoid arthritis and with other type of diseases. So that's why this pen, and I don't have it with me, sometimes I do, is really bulky. Is something that is very easy to use. You just take a cap, number 1 at the bottom. It is pressure sensitive. You then have another cap where you practically show a plum color activator button. You put that in your thigh, you press it, and then the entire biologic, in this case Humira, is delivered. This, and you see me about 20 years earlier, a little bit more hair than I have now, and we were considered really at the forefront of this indeed. In the next slide. In the next slide, you can see a paper that I practically worked or authored behind the scenes. You see me as the last author with a number of investigators. And we conducted a study comparing this device, the Humira Pen, with the standard injector, excuse me, the pre-filled syringe. And you can see that in this slide that when we look into ease of use, convenience, time of injection, safety, pain, and overall, it was a real smashing hit. And indeed, nowadays, all the companies with biologics are also producing these combination products. And that's a very important concept, because this a combination product is when you put together a medical device and a biopharmaceutical. And in this case, obviously, this is a combination that needs to go also through the FDA in that case, and also all regulatory agencies. We did that. I was working very closely with the regulatory team. These were other times. Now I have a very small team in my emerging biotech, and we do that almost the same things that we did in big companies, but through consultants and through vendors and CROs and a number of ways to do that. So my point is that we are joining by the hip, that medical devices and biopharmaceuticals very often come together. There are devices that are very clearly regulated by the agencies. And obviously, the first step is to, in many cases, or while we have this product in the market, we also want to make sure that we have a number of investigators that work with us. So this is the background that I would like to discuss with you. I must say that this work that we conducted, the Touch trial, was a lot of fun, and lots of citations for this paper, and many people still coming to me with ideas about how to develop pens. Next slide. So let me now spend a moment on this concept of personalized medicine. First, to the upper left, left. We have the traditional one size fits all. And that's the one example with the Humira. Nowadays is practically 99% of the patients receive that through the Humira Pen, the most successful biologic in history, and probably the more successful drug, still making billions of dollars for AbbVie. And all the patients receive the exactly the same dose. They practically, when a doctor decides, I want to give this drug, then they absolutely receive it. Although we know from our clinical trials, and I was also involved in clinical development of Humira independently of the pen, we also knew that a number of patients would not respond. And you can see that, but obviously what we present to the agency is the difference between patients that are responders and placebo. But there are a number of patients, which could be up to 40% of the patients that don't receive the drug. What do the doctors do with that? So they keep giving the drug until the patient starts saying, okay, I don't see any effect. And then you just move on. Now, we're moving into personalized medicine approach. That means that we now identify a signature. In this case, I refer here to the genetic profile. These patients would receive the targeted therapy. Other patients with other type of signatures would not, and would go into other types of therapy. Could be an alternative, and there are several alternatives to Humira right now, or it could be other type of drugs. And again, I'm using the concept where I'm familiar with in the targeted therapy space. Next slide. Now, as an immunologist, as someone, and when you work in that space for many years, you're also a cellular biologist, a protein biologist, and you are also a molecular biologist. So what I've realized is that there are several elements that need to be taken into account when conducting studies or clinical trials, and this is the so-called omics data. So we need really, and this is the definition from the FDA, biomarkers that provide information essential for the safe and effective use of a corresponding therapeutic product. And here you have different types of genomics data. It's not just genomics, transcriptomics, epigenomics, proteomics, phosphoproteomics, metabolomics, and probably there are many more. I've seen several others that are not in this list. But at the end of the day, it doesn't matter if you have a single biomarker or you combine all of them and create a signature which uses either big data, AI, machine learning, and at the end of the day, you decide very precisely which patients are getting your product. Again, could be a combination product. So that's the way we are progressing very, very rapidly, and there are many centers around the world working on that. That's what we have embraced at ASC Therapeutics. Next slide. So let me now start giving you. I gave you a kind of overview about how I see this field, everything that is taking place in the in especially in the therapeutic area, but obviously connected in many cases with combination products with medical devices. So next slide. Taking as an example hemophilia A. This is a very serious condition, is a monogenic disease carried by men, and this leads to patients that do not have enough clotting factor number 8 for hemophilia A. There is also the hemophilia B, where the missing factor is 9. And you can imagine this is a cascade, and of those of you that are also used to these types of cascades, it could be phosphorylation, it could be complement activation. They require to have all the elements, all the factors that if one of them is missing because of a genetic defect, then you get this very serious disease. So hemophilia A is again the lack of factor 8. And the majority of these patients survive over time because they receive either replacement therapy or something called bridging therapy. So they practically bridge the fact of that clotting factor with another, even a bispecific antibody is called. So what happens with these patients that need to receive these replacement therapy, sometimes blood infusions for life, or other type of synthetic factor 8s, they develop inhibitory antibodies. So we need to measure them. We need to make sure that we understand how these inhibitory antibodies potentially are going to impact the production of any type of factor 8. So that's why, for instance, we are working with a group in the University of Emory in Georgia, where they are realized that in cases where you have these inhibitors against human factor 8, you could use porcine factor 8 derived from the pig, but also does the same job. So there is lots of research in that space, and this is the group that we approached when we decided to go for gene therapy for these patients. Next slide. So I would like to use this slide to ensure that everyone in the audience is aware of the absolute revolution that we're going through in the development of biopharmaceuticals and, of course, associated devices. We started with small molecules. Obviously, we have still them around. We had large molecules. I was working in these large biologics. As you see here, could be compared with a jet fighter. It was a fun time. Other companies practically increased the complexity of this drug to make it more sustainable and durable in circulation with better pharmacokinetics and pharmacodynamics. But where we are now coming to, and this is probably the most exciting time in my career, is to gene therapy, where we can compare that with the International Space Station. It's extraordinarily complex the way we construct a virus that contains all the information we need to infect the cell and ensure that it delivers their payload, which is the gene. And by the way, everyone starts now to get more used to this because you are hearing about the AstraZeneca COVID vaccine, the Johnson & Johnson COVID vaccine, or the more developed, which are also related to these, the mRNA vaccines from Moderna and Pfizer, BioNTech, where what we have here is just the mRNA. So we skip the DNA and go directly into producing protein. So this is an extraordinarily exciting world. There are many devices associated to all these changes, and I want to make sure that when we approach investigators, that they are also aware and they can help us doing whatever work we need to do, either in the preparatory phase, during development, or on regulatory paths, and also after the phase 4 clinical trials. Next slide. So I like to use an analogy, and this is using rockets and modules and the lunar module here, which is the Apollo 11 historic journey, where we have a rocket that contain the promoter, which is kind of a capsule that had was leading practically and taking the lunar module to the moon. And in some way, that's what we have in place, and we as ASC Therapeutics have been developing. So we have a virus, which is the rocket, is AAV8, it's an adeno-associated virus, totally harmless. We use a liver-specific promoter. This is the capsule that is really driving all the way the journey into the nucleus, and then a specific transgene. And this transgene is very specific and different from anything in the industry right now. And I say in the industry because many other companies are also working on factor VIII gene therapy. But we have an engineered liver-specific promoter. We have an engineered liver-specific factor VIII transgene. And transgene means that the gene has been engineered in a way that is able, in this case, in our case, to be more efficient in terms of synthesis and secretion. Next slide. So the way it works is that we infuse millions of viruses into the individual, into the patient. It's just one single infusion. So it was one and done. And I'm right very much at the end of several years of work in our organization. In the next few weeks, we expect to submit an IND, and we expect to get IND clearance from the FDA. And in the next few months, we're going to treat our first patient. So we have already done all this. We have prepared the viral capsid with the transgene, with the promoter. Everything is in place. Now, what happens? Because this virus has certain tropism for the liver, it's going to go into a liver cell, is the hepatocyte, which we're going to use as a biofactory in some way. So we're going to have this virus entering the cell, then we are going to have it approaching the nucleus, and when it's in the nucleus, it's going to release the payload, and the payload is the promoter, the transgene, and also it's going to be walking in some or orbiting around the moon, and this is around the chromosome. So it's not going to be integrated, but in some cases we are able to do, and we have a separate program with the same transgene that is going to integrate this information into the chromosomes. So all this is extraordinarily complex. It requires a lot of know-how from us and from the investigators that are going to be helping us. Next slide. So when we enroll, when we speak with investigators to work with us in sponsored trials, in investigator-initiated trials, or when other type of, I would call them mixed or combined trials, first of all, we need to have an investigator that really knows very well the disease target. And they should understand how it works in terms of the actual factor VIII levels that are required to have factor VIII activity. So these are two components that we measure in our studies. So we want investigators that really understand that they also, when they incorporate patients in their study. And by the way, I'm talking now in general about our preparatory steps for an actual phase 1/2 clinical trial, which is very far away from investigator-initiated studies. But I can tell you, I'm very familiar with them. I've been there. I've been doing that with Abbott in phase 4 clinical trials. But at the end of the day, the mechanics are the same. You also need someone that really is going to help you in ensuring that you enhance the value of your device or biopharmaceutical. So we want to, the physician that is working with us at any of these levels is also able to understand the way he or she is going to measure a number of biomarkers that are going to help on inclusion or exclusion of patients, like inhibitory antibodies to factor VIII protein, as I mentioned a moment ago. Antibodies also against the virus, they exist, and a number of patients cannot even receive gene therapy because they have already in advance these inhibitory and neutralizing antibodies. Since we're using the liver to as a biofactory, we need to understand how the liver functions, and there is the liver enzymes, there's imaging, and then understanding how there are differences between patients depending on the type of mutation that they have. Some patients have a little bit factor VIII, so they have their immune system has already been exposed to factor VIII, others have not. And then the monitor, how we monitor biomarkers. So once the patient is in the trial, in the study, we need to ensure that we understand what type of biomarkers or what type of tests or imaging or are we going to utilize. And definitely, especially from the perspective of the regulator, safety is absolutely essential. And we want to understand that the AAV or our virus is really targeting the organ that we expect and not other organs and tissues. We also identify the antibodies. We look again into factor inhibitor levels, and of course, at the end of the day, efficacy is the key. So, what's the efficacy of our therapy? And in this case, is factor VIII activity. Next slide. So with that, I'm going to move just a few minutes into another example or case study based on my current experience. I'm skipping many other things I've been doing during my career. And this would be on cell therapy and how we select investigators here. And the case study is about graft versus host disease. Next slide. So here to the left is just a schema telling you about what are these deciduostromal cells. When you have a pregnant woman, they have the placenta, and the placenta is a number of membranes. It's a new organ indeed. It functions as such to protect the fetus and to provide nutrients and ensure that the fetus develop. This has been obviously part of the evolution of mammals, and it has also a very key function, which is defend and protect the fetus from the immune system from the mother, which eventually would recognize as foreign the antigens from the father. 50% of those in the fetus are should be coming from the other parent, and the immune system would eventually recognize them as foreign and would attack the fetus. So there is a way to protect that, and this is by the decidua. And in the decidua, there are some cells called decisional cells. They are a type of stem cells. They don't have a pluripotential capability, so they are very stable. We're able to have them in cultures for up to 5 passes, and they maintain their phenotype. And what they do, is, and you can see here, is that they increase the PD-L1, PD-L2, which is very well known in cancer. In this case, we increase that, decrease that, so that we are able to control and attract the T cells that we want. We increase the regulatory T cells. We control the acute inflammation on these patients, and then we apoptose reactive T cells. So what happens is that in patients with graft versus host disease, which is the disease that we are targeting here, what happens in this case is that we are going to avoid that the cells that are infused into patients to get rid of lymphoma or leukemias and so on also attack the organs of the individuals, of the patients. So in this case, we saw in graft versus host disease very little there, clinical ORR over 80% in AGVHD, which is acute graft versus host. This is a very serious condition, and it develops in a matter of days. So we're already planning on these type of studies. And next slide. Again, here we want to have investigators that are familiar first with the disease target, this steroid refractory acute graft versus host disease. So there are infusion centers, there are cancer centers that treat patients that receive bone marrow transplantation, and they are very familiar with this condition. There are ways to treat that. You always start with with the steroids, either those steroids, if they don't respond, that's when you need to do something, and that's when a cell therapy would be indicated. So it would be a second line type of therapy. In this case, we also have a number of selection biomarkers. We want to see how is the profile of circulating T cells and cytokines, practically understanding in general key elements of the immune system. There are certain tissue resident immune cells in the gut skin, which are the organs usually most affected by these graft-versus-host disease. And also we want to understand what's the difference between this treatment and other treatments. The investigator should be able to monitor biomarkers, and especially, and that applies across the board. It doesn't matter if you have a device, if you have a biopharmaceutical. The regulators are going to be interested in safety. You can have safety related to tolerability of the device, but at the end of the day, is safety related to the actual drug and how it works. So you need to predict with these multi-omics that I mentioned a moment ago, the immune-related adverse events. You need to also immune profile these patients, potentially with a new technology called mass cytometry that we're using. And also, we want to see the efficacy of these therapies. And here we have something called the magic biomarkers, which were developed in Mount Sinai, and they are already accepted by the industry. So we need to ensure that we understand the entire field and we know exactly what type of biomarkers we want to be monitoring. With that, next slide. I just would like to indicate how important it is that these investigators have certain key traits that they improve the understanding of the biological the aspects of the biological and genetic drivers of the disease. They need to understand that we are now moving into next generation sequencing and computational biology and big data. So that's the way you put together all these data together, are able to produce predictive models of gene expression, regulation, and so on, and understanding the complex mechanisms of diseases. Thank you so much. And next slide. Yeah, again, thank you. And just for all of you that are interested in this area, I would like to suggest that you also join the Medical Affairs Network in LinkedIn. I'm the manager of that one. It has close to 20,000 people, and we talk all the time about investigator-initiated studies.
Great. Hello. Thank you so much, Oscar.
Thank you.
Thank you. Thank you very much. I'm just going to, before you go, I'm just going to throw a few questions at you, if that's okay. What is the role of investigators in combination products of drugs and medical devices?
I think that the investigators have a similar role if it's a medical device, is a drug, or is a combination product. And at the end of the day, it's all about having someone that, in my experience, has to be motivated to work with you. And I've been working with many investigators, and they have to have it inside them. They believe in your product, and they are going to be the ones conducting your study. So absolutely critical, I would say, interest, motivation, and know-how.
Okay. So what is, I'll go for this one. What is the level of know-how required by the company medical teams for gene therapies involving medical devices?
Very high.
Very high.
So I can tell you that I would suggest anyone trying to get into this field, which is going to be revolutionary. It's already happening. There are over 500 companies working on gene therapy right now. People don't know about them because there are just a few products approved. One of them is Zolgensma. Well, I was also involved with a company called Avexis, acquired by Novartis. So you need to understand everything related to these biological processes taking place. And by the way, if you just read about mRNAs and DNA vaccines, you're going to learn a lot. So the information is out there. Go into that and ensure that you follow all the experts in the field.
And just in terms of submissions, should study investigators be involved in regulatory submissions of gene and cell therapies?
So, I mean, it's a little bit the chicken and the egg. So at the end of the day, you want them to be involved with you as soon as possible. Because I've always been the same with my entire career. I go for the best in the field. and I look into the country where I'm planning to have regulatory trials. I select them. I start working on creating publication with them, which is very motivating. I work with them in the regulatory submissions. But whenever I get into phase 3 or and then especially phase 4, I plan to continue working with them. So it's all about relationships. You have to initiate these relationships in the regulatory submission time, and then moving them into the different phases with them, holding hands with them in some way.
Great. Thank you so much, Oscar. Thank you for joining us. That's fantastic. Thank you for closing the day of the speakers as well. Thank you.
Thank you.